Welcome everybody. Good morning, afternoon, or evening, depending on where you are in the world. I want to welcome you to this, which will be our last lecture in our microplastics series. And since it is the last in the series, I want to take the opportunity to thank all of you who have joined us faithfully every week. I hope you've enjoyed the series. I know I've certainly learned a lot from it. If you have suggestions for future lecture series that you would like to see us organize, by all means, please get in touch with either myself or Raquel. And I want to take a minute to thank Raquel for all of her work associated with organizing of the lectures that we've had in this series. So today, I want to welcome Dr. Joana Prata. Joana is an assistant professor at the Abel Salazar Institute for Medicine and Biomedical Sciences of the University of Porto and the University Institute of Health Sciences of the CESPU in Portugal. Joana holds both a Doctor of Veterinary Medicine and a PhD, and she's an invited assistant professor at the Abel Salazar Institute. She received her degree in veterinary medicine in 2016 from the University of Porto, and is certified by the Portuguese Veterinarians Board. Her PhD in biology and ecology of global changes with a specialization in environmental biology and health was received in 2021 from the University of Aveiro with merit and distinction. And today she's going to be talking to us about some of the human health implications of microplastics. So welcome, Joana. We're very pleased to have you here with us today.
Thank you so much for having me today. I'm going to share my screen. Let's see if this works. So today we're going to talk a little bit about human health implications of all that we've been talking about in the past seminars. As I've been introduced, my name is Joana Prata. I'm a university professor in Portugal. I spent the past, I don't know how many years, but almost a decade studying microplastics. So I was able to accompany this issue from almost the beginning of the research. So I hope to share today with you all my experience in working with microplastics, especially regarding the human health implications. So I think the first thing, probably you're sick of seeing slides like this, so that we can contextualize why microplastics are a problem as well as plastics. Well, we have to look at plastic consumption. And as you can see in this graph below, the world consumption of plastic is still increasing, while in Europe is starting to stabilize. In Europe, because we have a lot of regulations about the use of plastic and plastic waste, so there is a bit of stabilization. But why is such a great demand for plastics? Well, plastics were introduced as a miracle material because it was lightweight, transparent, flexible, low cost, and it did a lot of great things for us. In the case of medicine or food safety, it is essential in these areas. However, we started using them to substitute other materials in other areas and to produce a lot of waste. And as you can see here on the slide below, most of plastic is used in packaging, almost 40% of plastic. And this packaging, while it's great for food safety and all, it's not very good in terms of end of life, because most plastic is still not recycled. And so it ends up in landfills. Some of it is released to the environment accidentally. And so we have all these plastic waste to deal with. Then the second category is building and construction. This one is not as bad because the plastics we use in buildings usually stay there for a long time. Then we have automotive, which is also a long-term application. And then electric and electronic. This sector is growing a lot because all of us are full of electronic gadgets in our lives, including our smartphones. And so we are generating a lot of electric and electronic waste. And all these equipments have plastics in them. And these plastics are full of additives, so they don't burn when we use the equipment. And because of the electronics, there's a lot of metals associated. So the plastics and microplastics coming from electrical and electronical waste are likely to be more toxic than the others. So regarding microplastics, we have these plastic particles below 5 millimeters, which we can produce them under these sizes. and they can be produced in nurdles, as you know, the ones used by the plastic industry, or in products such as cosmetics. Then we have secondary microplastics, which come from the fragmentation of materials. And of course, we always think about these fragments we see on the beach that comes from all the plastic packaging that found its way to the environment. But actually, this is happening in every plastic object. Their surface in your room right now, you have plastics, of course, even your laptop and your phone, and they are releasing small microplastics to the air, as well as our clothes, which are also made from synthetic materials. So these microplastics, of course, these big ones in the beach we can see, but there's a lot of tiny ones that are even in our houses, in the air we breathe. So they are prevalent, and they have been found in every environment on Earth. The cycle of microplastics, you're probably already aware of this, but we have it's produced mostly from oil, and it can be used as microplastics in applications such as cosmetics, but also when we wash our clothes, we release microfibers. And this goes into the wastewater treatment plant, which retains most of the microplastics, but some of them, even with a 99% efficiency, a lot of microplastics will be released. Why? Because we're talking about millions and billions of particles, and even 1% of that, it's a lot of particles being released. Then we have the other pathway, which is from the fragmentation of objects, and all these microplastics find their way into the environment and eventually into organisms. And we humans, as top consumers, we are exposed to these organisms contaminated with microplastics. As you know, everything is contaminated with microplastics right now. Most studies have been focused on the aquatic ecosystem because that's where we started seeing microplastics for the first time. But now we know they are in every type of environmental matrix. They are in the air, they are in the soil, in the sediments, and they circulate between these different matrices and interact with the organisms living there. So we can have ecotoxicological effects directly on the organisms. They can impact the ecosystems and impact geochemical cycles, as well as abiotic properties of the environment. For instance, in the soils, they can change the soil permeability to water, and this can have a great influence on agriculture as well. But microplastics do not only have this effect based on their particle, on the particle effect or the leachates they release. They can also be vectors for microorganisms. Actually, microplastics are a substrate that is being contaminated, being taken over by microorganisms in the environment. And the ones growing on the plastics are different from the ones growing in other environmental matrices, for instance, in wood or in the soil surrounding the microplastics. So it creates a niche environment for microorganisms, and they can actually increase the persistence of these microorganisms in the environment or even transport them long distances, because while wood will degrade, for instance, microplastics will not degrade as easily. So they have a lot more potential to transport these microorganisms. Then they can also act as vectors for chemical contaminants. Depending on the environment, on the microplastic, and on the contaminants, sometimes contaminants can adsorb to the surface of microplastics. Of course, as you know, as any chemical interaction between particles and chemicals and contaminants, it will depend on the environment surrounding the particle and the contaminant. For instance, the pH can influence this relationship. But our concern here is that microplastics, along with contaminants already in the environment, can have a synergistic effect on the toxicity they have on organisms, but they can also act the other way. Microplastics can actually decrease also the effect of the contaminant if they absorb it so strongly that it's no longer available to the organism. So it really depends on the conditions this happens. Of course, it can also increase the toxicity, which is why we are worried about it. So there's a lot of challenges in working with microplastics. Generally, microplastics are a class of contaminants. Of course, we have been taught to think about microplastics as a contaminant, But in itself, it is not. It is a class because each particle is unique. They have different properties such as size, weight, volume, chemical composition. So at each time you find a microplastic particle, it will be completely unique because that size, that shape, that chemical composition, the absorption of contaminants from the environment, the microorganisms living in its surface, and so on, will be completely unique. So we have to talk about the class of contaminants. We cannot replicate this individuality in the lab. It's impossible. What we can do is study each condition, how it affects the toxicity and the behavior of microplastics. For instance, just looking at size, well, you know that most studies have used the size limit of 333 microns, which is the mesh size of a manta troll, a net that is used to capture microplastics from water. And even though most studies have used this size, if you look at the studies considering sizes above 1 micron, up to the limit of 5,000 microns in both, of course, the limits of which we consider a microplastic, you can see that looking down at 1 micron particles, we find 40 times more particles than just looking at the ones that are above 333 microns. So the size, just deciding on what size we will look for in our study can have a big impact on the results we find. And this is just talking about the size. Of course, all variables that you select in your methodology can affect what you will find. So this is a great challenge also to compare studies. And of course, the ones we see more often on tissues of organisms inside the organisms are the smaller size, the sizes below 20 microns, which is obvious, I think. The smaller particles are more likely to move to internal tissues. And so these are the ones we find more often on the tissues of organisms. However, it's the ones that we find it more difficult to identify in the lab because these very, very small particles are difficult to see, even in a microscope, and to quantify. But we have to focus on this small fraction if we aim to understand the impacts on organisms and human health. So the big question is, are we exposed to microplastics? Well, you already know this. Of course we are, and there's two big pathways for our human exposure. It's ingestion and inhalation. Of course, dermal contact could also contribute, but in the case of microplastics, because if we consider microplastics ending at 1 micron in size, and then below 1 micron it's nanoplastics, well, in the case of microplastics, they are too big to cross the stratum corneum of our skin. So in this case, they don't seem very relevant as a pathway of exposure for microplastics. If we go down to very small nanoplastics, maybe even the skin can be a route of exposure. But for micro, we think about ingestion, what is in our diet, and inhalation, what is in the air we breathe. So for the ingestion, we have here a study a student of mine did, which basically she collected information from the literature about fish, and this is mostly the presence of microplastic in the gut of fish. And we see here that most fish contain microplastics in their gut, around 60% of fish globally that already contain microplastics in the gut. And the carnivore species presented more microplastics than the omnivore species. And you have here the map. You can see that most circles are the big ones, and the orange and red ones, which means that a lot of fish are contaminated, even in different areas of the globe. And we eat this fish, so we are worried. Oh, sorry. And we actually went to fish tissues to look for the microplastics in there, looking down to sizes above 1 micron. And in here, our purpose was not to look for the exposure, the human exposure. So we did not sample fish muscle, which is what we consume when we are eating fish. We looked at the internal organs of fish because we were interested on where the microplastics were going inside the body. So we have the gills of the fish, which is a very highly exposed tissue because a lot of water is running through the gills. And so we found there a lot of plastics. And we have the heart. This fish we use, Tracurus, is usually sold very small here in Portugal. And so it's very difficult to get a good amount of blood to test for microplastics. So instead, as a representative of the circulatory system, we use the heart. And as you can see in this picture, the microplastics that we found in the heart were generally very small, as we were expecting. And then we have the kidney, just to show they were moving around the tissues, including tissues related to excretion. And then in the gut, we found the largest sizes in the gut. This is also because we included not only the tissue of the gut, but the content in the gut. So some of these plastics could actually still be in the lumen of the gut and not on the wall. And as you can see here, as we mentioned previously, most microplastics are below 10 micron in size, so very, very small. And these were suspected microplastics that we saw with Nile red, because these very, very small plastics are difficult to detect with other techniques. Even if we go to microrama or micro FTIR, these sizes are too small to get a good reading. And often they fly off when we hit them with the laser to get the excitation light. And so it's very difficult to get a good signal from them and confirm them. But they were Nile red. Using Nile red, we saw them tagged. And then I tried to aggregate information about the exposure through consumption of animal products, but from terrestrial ecosystems, because we have a lot of information about the fish, because there's a lot of studies focusing on the aquatic ecosystem, but ecosystems are still understudied. And here I tried to get information from different types of foods, of animal products from terrestrial environments, and I used the daily intake proposed by the World Health Organization and the European Union trends to calculate what was our exposure through the consumption, the normal consumption of these products. And as you can see here, we have a lot of missing values for a lot of different types of food products, such as liver, kidney, animal fat, pig meat, bovine meat. This is all understudied. And even with all these knowledge gaps, and even considering that the studies used here, the literature I've used and collected from what has been published, has used different methods. So we can have that problem from the different sizes used and all the chemicals being different, having contaminations, the contamination control not being equal between all studies. So we have a lot of variability and a lot of missing values. But if you look at the estimate, the worst case scenario is that we are daily exposed to 15,000 microplastics per day, which is a lot. Of course, not all of them will be absorbed by our bodies, but still it's a number to take into consideration. And we really need these numbers of exposure, because without them, we cannot conduct risk assessment. And the problem is that there is a lot of factors involved with the microplastics. Of course, the animal itself can be contaminated in vivo, when it's still living its life, it's exposed to microplastics, and microplastics go into the tissues. But there is also some contamination during packaging and preparation. So the plastic cutting boards, these studies found that they contribute to microplastics in chicken meat and goat meat. So just cutting your meat over a plastic cutting board can release microplastics to your food. And I think this is a little bit obvious, because we can see when the cutting boards are getting old, we can see all these fissures caused by our knives. And basically, every time we do this, we are releasing microplastics into our food. Then we also have the packaging. Well, we have chicken meat packaged in extruded polystyrene trays, which also release microplastics, takeout containers as well, and just opening a plastic packaging produces microplastics as well. So all these are sources of contamination beside the the contamination already present in the animal when it was alive. And for water, well, our water courses are contaminated as well. Here are results from Aveiro Lagoon and Voga River in Portugal. Voga River had a very low concentration. Also, we were looking for microplastics above 50 microns. And here the concentrations were low because we went into in the countryside with little influence from anthropogenic sources. But if we look at the Douro River, which is near Porto, Porto is the second biggest city in Portugal. And you can see here, the river is flowing from the right to the left of the picture. And if we go to the countryside, more on the left, we found very low concentrations of microplastics, fibers, and synthetic fibers. Then if we sample closer to the city near a wastewater effluent release, we can find higher concentrations of microplastics and fibers. And if we move towards the city near a boat repair shop, we find even higher concentrations of microplastics. Of course, here originate from the boat repair station, or just because we are moving towards the city and towards the estuary and the concentrations are increasing. The presence in wastewater is also a little bit concerning because it means that microplastics will also be found in the sludge and biosolids. And these sludge and biosolids, we want to use them because they are rich in nitrates and phosphates, which are fertilizers. We want to use them in agricultural soil as a fertilizer. And if they are carrying microplastics as well, it's bad news because we are contaminating the agricultural soils. But not only the water in the rivers is contaminated, our tap water is also contaminated, as you can see here. For the tap water, 10 microplastics per liter of contamination. But if you're working in the lab and using ultra pure water, you can also find microplastics in it. So you have to filter everything when working with microplastics. And the ultra pure water, if you read the details, the technical details of the machine, you will see that some level of particles is accepted as part of ultra pure water. And some of these particles will be microplastics. So we really need to filter everything, and even the water in our homes is contaminated, not necessarily only bottled water in plastic packaging. But wine is contaminated as well. So you really have no way to win this battle against microplastics. So we also have found these microplastics in wines, and we confirm them using micro-Raman spectroscopy. So what happens when you eat this fish, this meat, or drink the wine or water contaminated with microplastic? Well, it will end up in your gastrointestinal system. And most of it, the higher concentration of microplastics, will be in the lumen of the gastrointestinal system. Then we have a mucus layer that covers our cells, and only a few particles will be able to cross the mucus layer and get in contact with our cells. Well, when it's in contact with our cells, it can be internalized in a lot of ways. We previously thought that M cells were the major entry point for microplastics, but actually looking at these old studies where they have conducted a lot of exposure to microplastics. At the time, they were not called microplastics, but studies from the 70s and 80s where they called them latex beads, but basically they were microplastics. They found that the M cells were not such relevant source of entry as the enterocytes. This means that most microplastics that can cross the mucus barrier, some of them will get through into the enterocytes through transcytosis and will reach the internal tissues through the enterocytes. Some of them will also be captured by macrophages, and some will be transported between the cells by paracellular transport. And these microplastics will find their way, some of them, very little, less than 1% that we ingested originally. They will find their way into the interstitial tissue. And here it can either go into the Peyer's patches, it can stay in the tissue, it can go into the Peyer's patches where it can be internalized by macrophages, or it can move into our blood vessels and be transported across the body. But the air is also contaminated. So we have another point of entry. Well, these are fibers that have been deposited in filters. And of course, this is under the UV light. So you can see the fibers, depending on the pigments used in the production of the fibers, or some fibers may have a natural fluorescence. You can see them in here in these filters. And this is actually also happening where you are. There are fibers in the air, and the heaviest ones and largest ones will get deposited in your surfaces. And so the microplastics here in our laboratory, which had a very strict cleaning schedule, was based in 6 particles per hour. And of course, we work inside the laminar flow hood to prevent contamination of our samples. And in this case, you can get less than 1 particle per hour. But this means that every surface in your home is covered with microplastics. And even when you're eating, there are microplastics coming from the air into your food. So we looked, this is a first attempt at looking at the microplastics being deposited in different areas of the university. Because we did the sampling only for 4 hours, because we wanted the use of the space to be uniform during the sampling time, we found very low concentration of fibers, and most of them were not different from the blanks. The one that was different was the library, probably because there's a lot of people going in and out of the library, and also because of the surfaces, which are very difficult to clean. And in this case, we estimated the deposition rate of fibers of 216 fibers per hour per square meter. Of course, some of these fibers were synthetic, not all of them. And the ones that are synthetic can be considered microplastics as well. But the good news is that the ones we found, most of them were under larger sizes, above 100 micron. And this means that these fibers will not be able to get into our lungs. We also did an active sampling using an air pump in Aveiro, in indoor residential environments. And we found relatively low concentrations of microplastics and synthetic fibers, about 1.1 synthetic particle per cubic meter. And you have here some examples. Of course, most of the particles were very small, and we had some fibers, both synthetic and natural fibers. And we have an ongoing work where we are trying to identify the microplastics from nasal lavage of participants. So we ask people to inject a little bit of water into their nose and collect it into a tube. And we look for microplastics in there, which means that the person is breathing in these microplastics and it got stuck in their nose. So we also had a problem with our blanks having a lot of contamination. But we can see that some of them, we actually, after even subtracting the blanks, we found a lot of particles here. For instance, the person 9 had 96 particles per 12 milliliters of water injected in the nose. So we can also find the microplastics in our bodies. So what happens when we are exposed in the air to microplastics? Well, we have some clearance mechanisms in our body to deal with particles, because naturally we are exposed to particles. You can think of, for instance, sand. We are exposed to sand naturally. So our body has developed some mechanisms to deal with this. Some of them are mechanical, and this means that when you breathe in a lot of dust, you cough or you sneeze, and this will make the particle leave your body. Then in the airways, we have the mucociliary escalator. Basically, we have cells which are beating, we have cilia beating, and these cilia are moving the mucus on top of it. And they are moving this mucus towards our throat, where it will be swallowed. So the particles get into the airway, they can get stuck in this mucus, and then they will be carried in the mucus towards our throat, and they will be swallowed and find their way into our gastrointestinal system. Of course, if the particles are small enough and can find their way into the lungs, they can be, the macrophages can clear them out, or they can be transported by the lymphatic vessels. And the particles, it will depend on the size, the shape, the way that they can deposit in our respiratory system. So the ones that can find their way into the alveoli, a lot of things can happen here. And basically, this is a representation of the alveoli, and only very small particles will reach this stage, generally particles below 5 microns. And here, again, it can be internalized by pneumocytes type 1 and type 2, and macrophages that are clearing the alveoli. Some of them will get internalized as well between the intercellular spaces, and the internalized particles in the interstitial tissue can stay there or can move into the capillaries to reach the systemic circulation. Some clearance will also occur because of the anterior ciliary movement, as we talked about, and the lymphatic drainage, but the macrophages will also clear a lot of them. And we found them we find these particles in the lungs. You can actually find particles above 5 microns in the lungs. This does not mean that the particles came from you breathing in the microplastics, because some of them will just be circulating in your blood and getting stuck in the small capillaries of the lung. So the larger microplastics that might gotten into your blood from the gut, actually, when it reaches the lung, they can get stuck in the small capillaries, and then you will find larger particles in the lungs as well. But are microplastics found in human tissues? Well, you actually already know the answer to this, of course. Yes, they have been found in the lungs and airways, liver, colon, kidney, spleen, blood, placenta, and feces. Feces, most likely because it's the main excretion pathway for microplastics. So these are some recent publications on microplastics. I thought these were very interesting. Once this study found one microplastic in a thrombus, and this one found microplastic in the human blood in concentrations of 1.6 micrograms per milliliter, which is actually a high concentration, in my opinion, because usually we are talking about nanograms of contaminants in the blood, but here we are talking about 1.6 microgram. And you can see here that the different people had different compositions of microplastics in their blood, and some people had even higher concentration. In our case, because the human samples are very hard to come by and to deal with, and because I'm a veterinarian and part of the veterinary school here in Portugal, we actually have animals that come to the clinic, and when they die, sometimes the owners ask for a necropsy evaluation, so we can determine the cause of death. Take into account, we already had these animals here that died naturally or during the clinical treatments and so on. We actually, during the necropsy evaluation and not compromising its results, we gathered materials, biological materials from these animals. And we collected samples from the lungs, heart, the heart to collect the blood, and kidneys and liver of these animals. And we found that 35 out of 49 animals had microplastics in their tissues. And in this case, we found them using Nile red staining as well. And they were present in 80 out of 242 samples. Again, most of the particles were 1 to 10 microns in size, so they were very, very small. And actually, we got some of them characterized, and you can see here, for instance, we found polypropylene and polyethylene terephthalate in the lungs of a dog. Well, actually, companion animals like cats and dogs are very good to estimate our exposure to microplastics because they live in the same environments as we are, and they can be considered sentinel species. Of course, there are some things that are more appropriate to estimate from the animals than others. For instance, for cats and dogs, the airborne exposure is very likely to be similar to ours. Of course, we have to consider that they are shorter, they are closer to the soil, so their exposure may be higher to these particles because there is some resuspension of particles that have been deposited in the soil. And so they may be good sentinels for airborne exposure. But for dietary exposure, maybe not so much, because of course we sometimes share our food with our dogs and cats, but most of the food, it's pet food. So their dietary exposure is not really similar to human dietary exposure. We also have tried to look for microplastics in other model organisms, for instance, in a planaria. And you can see here this experiment with planaria that were fed live prey composed of Chironomus riparis. This is a mosquito larvae. And this mosquito larvae was previously exposed to polyurethane microplastics. So the larvae ingested the polyurethane microplastics, and then the planaria ingested the larvae. And we saw that the microplastics got into the planaria through the food chain. And we tried to analyze them by histological analysis, and we found some suspected particles in the pharynx, as you can see here. And I brought this study exactly because of this. The histological analysis is very important to understand where microplastics are in the tissue, because most of these studies that I have found microplastics in tissues have digested the tissue. So we don't know if it was in the capillary, if it was in the interstitial tissue, or if it was inside one specific cell type. We only know it was there. So in the future, we need to have more histological analysis, but we have to find a way to make it work for microplastics, because as you can see here, it's very difficult to detect them in histological analysis. And so what are the consequences of human exposure to microplastics? Well, we already have talked a little bit about how we absorb the microplastics. We think that most of it occurs through the digestive systems, through the transcytosis in the enterocytes. Of course, some exposure may also happen in the lungs when we have these very, very small particles that we are breathing in. The distribution will occur through the circulatory system. And if the particle finds a capillary bed that is smaller than its size, it will become entrapped. And this happens especially in the lungs. For microplastics, we do not expect a lot of metabolism. If you have a plastic prosthesis in your body or any other kind of plastic, it will result in biodegradation of that plastic as a result of the physiologic fluids, microbiota, and the inflammatory reaction. In the case of microplastics, it is expected that they will be excreted more quickly than this metabolism acts over them. And then the excretion. Well, most of microplastics in circulation will get stuck in the spleen or in the liver, and they will clear out these microplastics. This clearance will be through the biliary ducts in the case of the liver, and through macrophages that capture these particles and come into the intestinal wall to release the particles back into the lumen. So most microplastics will be released into the gastrointestinal system again. Some of them will get reabsorbed, but many of them will get excreted in the feces. And this is why we find so many in the feces. In the case of the airways, we can look at studies from the 70s and 80s where there were no occupational safety rules. And so people were exposed to microplastics from their jobs because they were working with plastics or with textiles, which release a lot of microplastics, and they were breathing all that in because they did not have any safety equipment nor adequate ventilation. And they got exposed to very high concentrations over a long period of time. In these cases, we found that people often developed occupational diseases, cursing with dyspnea, and inflammatory responses. And this is likely caused by the dust overload, which is basically a positive feedback loop towards inflammation due to the presence of a lot of particles in the lungs, then oxidative stress, and then translocation into other tissues. If we look at the whole body, all the things that the microplastics can cause in the body, and they may be caused by oxidative stress and cytotoxicity, and this can be due to the high surface area of the microplastics, the release of leachate from the microplastics and by promoting inflammation. Then we have some changes, some disruption to the energy metabolism, which may be through the increased consumption of calories, through inflammation. In the case of smaller organisms, they can decrease feeding, and they have gastrointestinal effects. In our case, of course, our gut will not get filled with microplastics to stop us from eating other things, but the microplastics can interfere, for instance, with our digestive enzymes, so we cannot digest as well the nutrients. And we have some effects in immune function through the modulation. This means that it can promote or depress immune function, and can also be involved in autoimmune diseases, because if microplastics come and damage a protein, and this protein now has a weird shape or is somehow damaged, the immune system can detect this protein and start a reaction against it, and then basically it will start an autoimmune reaction. And then we have some effects of neurotoxicity, which have been mostly seen in animals, which have shown behavioral changes and changes in neurotransmitters, and this through the pro-inflammatory cytokines that are released due to all this inflammation caused by microplastics. Overall, there is this study I recommended. You have here below the DOI of the study. It tried to look at the molecular initiating event for microplastics, what was causing all these changes, and they saw that the reactive oxygen species is the likely initiating event for all of the other things we've talked about right now, for the inflammation, for the metabolism, for changes in behavior or neurotoxicity, cell death. All this seems to be related to the reactive oxygen species generated by microplastics. And then here is a work I did in ecotoxicity, just to show you some things that we have to consider regarding microplastics. First, it's very difficult to work with very low concentrations of microplastics that are representative of what we are being exposed to or what the organisms are being exposed to. So keeping this in mind, we have used environmentally relevant concentrations and also some high concentrations of polypropylene and polystyrene, both virgin and weathered. And we tested this in microalgae and in zebrafish embryos. The size of these microplastics were too big to get into the embryo or into the cells of the microalgae, but even though they were too big, they still exerted some effects. So we suspect that the effects were from the leachates that were released from the microplastics. In microalgae, we found growth inhibition and both growth promotion as well. And in zebrafish embryo, we saw a decrease in heart rate and a decrease in body length. Of course, these effects, and I think this is the important part, did not follow a linear curve of toxicity, which means that depending on the concentrations of microplastics, you cannot predict from one toxicity point, you cannot predict what will happen in another, because it's not following a linear curve. And I personally think that this happens because microplastics, depending on the concentration, they will tend to aggregate together, and this can influence their toxicity. And we also looked at microplastics in Chironomus riparius, which is a mosquito larvae, the one that we just talked about in a previous slide. And you can see here that we found short-term and long-term toxicity. And here, what is, I think it's most interesting, is that microplastics caused an increase in the mean time of emergence. So the larvae emerged later than expected, and this caused a greater gap between the emergence of females and males, which means that the mating of these organisms may be compromised. Overall, microplastics can cause a disruption of ecosystems and ecosystem services. So this means that the toxicity and the effects on human health are not only direct effects. We can also have indirect effects through the disruption of ecosystems and ecosystem services that we depend upon. As we talked about the species living on microplastics, which can be called the plastisphere, they can actually increase the dispersal of invasive species, and then changes in habitat properties. As we talked about, change in soil permeability can influence agricultural outputs. The thermal diffusivity can influence the sex determination of reptiles, for instance. So we can have turtles just being born all female or all male, depending on the heat in the sediment where the egg is. Then we have also effects on plants that we did not talk so much in detail, but the effects we talked about in organisms, in animals, can also be found in plants, which can compromise agricultural outputs again. Then we have effects on the nutrient flux and the biogeochemical cycles. And as we talked about before, they can be vectors for microorganisms and contaminants. Overall, they have effects in almost everything, and we really need a One Health approach to this. We need everybody to be involved, all areas, so we can address all these problems caused by microplastics and also the plastic itself. So this is the end of my presentation. If you are working with microplastics, I invite you to submit to my 2 special issues. One is about the animals as sentinels for microplastics, and the other one is just about monitoring microplastics in the environment. Feel free to talk to me about this. You can send me an email. You have my email address, and I hope that this presentation was useful to you. Thank you.
Thank you very much, Joanna, for an excellent presentation. Lots to think about in what you've given us today. So I'm sure that Joanna would be happy to answer questions. If we have questions from anyone, you can either open up your microphone and your camera if you wish to ask a question, or you can put it in the chat function.
Can I ask a question? But my internet connection is not so good. Yeah, so apologies, my internet connection is not so good, but I'll try. Joanna, great talk, really interesting. And so I understand, of course, microplastics are absolutely everywhere. And I guess the main question I have is then, how concerned should we be in terms of potential human health effects? And I think you brought up something very interesting about factory workers. And has anyone looked at factory workers? Are they prone to certain illnesses that may be related to plastic exposure?
Thank you, Julian. It was very interesting, your question. Well, how concerned should we be? In terms of acute effects for the general population, I think we should not be too alarmed. Well, we are here alive, all of us, and we are exposed to microplastics right now. So in this case, I think it's not so worrisome. What worries us is the chronic effects, the long-term effects, especially in the presence of other contaminants, right? We now talk about the expososome, the exposure to all these chemicals simultaneously. We don't even know which ones, and their synergistic effect over the same pathways. And I think the future will be about this and about the chronic effects. Then you were asking about the workers. Well, in that study from the year, I'll try to go back. Here, this study, I tried to gather the information from the 70s and 80s that has been published on occupational diseases from these areas where microplastics were present. Again, of course, at the time, they did not call them microplastics. We know this because the materials were plastic. And so here they found dyspnea they found acute effects in the workers because, of course, they were exposed to very, very high concentrations over a long period of time. And this was confirmed because some workers went home because of the disease. They stayed home and they got better. And then when they come to work again, they got sick again. And they could actually find the relationship between these two things, the inflammatory responses causing these symptoms and the exposure in the workplace. Of course, right now, I think things are a little bit better because people tend to use more protection. Workers now have masks and we have ventilation systems. We are a little bit more aware of this. So right now, I don't know about the occupational diseases being studied at the moment. I think it would be interesting, for instance, to go to waste treatment plants and also sample the workers there. But this type of exposure, like it happened in the 70s and 80s, it's unlikely to happen again, because we are wearing all these protection equipment. Hopefully it will not happen again, right? That people can stay healthy. But I think it would be an interesting thing to follow over a long period of time, to follow these workers who are more exposed than the general population. It's a good idea, Julian. If you want to pursue it, I think we would be very happy to have those publications available in the literature.
It's on my to-do list. But I think the difference between the historic work and maybe nowadays is that there may be a greater exposure to smaller and smaller plastics, whereas I think the historic work may have focused on larger plastics, I'm guessing.
Not necessarily, because these workers, you can imagine that in a textile industry, if you're cutting textiles, you're releasing a lot, if these are synthetic, you're releasing a lot of small particles as well as bigger ones, but the smaller ones are the ones that will get more easily into the lungs and cause these diseases. And so, or even if you're at home or in your work cutting plastic boards, you're also producing a lot of microplastics, even smaller ones, even though at the time they were not aware of it. So I think here the exposure might have happened as well with the smaller microplastics, because anytime you cut plastics or you break a plastic, you're producing microplastics. It is not necessarily the exposure to the sun. Of course, the sun accelerates the process, but the plastics just existing is slowly releasing microplastics. So in this case, handling the plastics in work, right? Because they are cutting, poking holes, or cutting tissues, cutting textiles, they can produce a lot of microplastics as well, even in the smaller sizes. And to get into the lungs, it actually has to be smaller sizes. Otherwise, what people would get is a flu, right? If the nose, and they would cough a lot and they would sneeze, but these larger plastics will not get deeper into the lungs. To get into the lungs and cause this interstitial disease, it must have been smaller microplastics. Just as a particulate matter, also only the smaller ones get into the lungs.
Thanks.
Thank you.
Do we have other questions for Joana? I have a two-part question for you, Joana. I was particularly struck by your comments about particles in the circulatory system that can ultimately get stuck in capillaries in the lungs. For an old person like me, it might not be such an issue, but for young people who, given what we know is in the environment, presumably are going to be exposed to small plastic particles for the extent of their life, do you anticipate that we're going to see more and more respiratory issues associated with plastics if over the course of a lifetime you're continually getting little particles stuck in these capillaries?
Well, I'm not really sure if you're going to see more respiratory disease. What we know is that they get entrapped, but eventually they will be cleared out. The macrophages will come, collect them, and release them back into the intestinal tissue. Of course, this will take a little bit more time, but eventually they will be cleared out. I think it's a bit worrisome, and I think it's a bit of a confounding effect, because we can collect tissues like we did with the dogs and the cats. We can collect tissues from the lungs, but if we don't look at the histology, we will not know if the particles are in the blood or actually the exposure is coming from the air. So it's very important to develop these methods so we can know where the particle is exactly, and if it's causing an inflammatory reaction or not. That's why I brought these slides here on the front about histologic analysis, and how difficult it is to find them. Because what we did in this study with dogs and cats is what most studies do, is basically we digest the material to extract the microplastics. So we don't know if it's in the capillary, if it's in the interstitial tissue, or if it's still inside the alveoli in the airspace. I think this is very important to determine what is the impact, the real impact of microplastics, and also to determine, as you said, if there will be or not an increase in respiratory illness.
So the second part of my question really, and it's one that's rather difficult, I know, for you to answer, but we have evolved in an environment, for example, where there's lots of dust, right? And so our bodies do have mechanisms to try to reject dust particles. Do you think that the mechanisms that are in place are capable of handling the microplastics? Or do you think ultimately humans, if we're going to survive, will need to evolve different mechanisms for eliminating microplastics?
Well, evolving new mechanisms will be very hard because it would have to have a big effect, so we can select individuals which survive. And most of us, since this is chronic, most of us are likely to have reproduced before. So selection, evolutive selection, I'm not expecting. But yes, the mechanisms we have evolved, these mechanisms of clearance, they were evolved for dust particles, for virus, bacteria that are getting into our airways. And this also act on microplastics. What is the difference? The difference is microplastics are very low density particles. Sometimes they are fibers, which means if they go a certain way, right? If they go like this, they can get through. If they go like this, they will get stuck. And also they are synthetic, so it's very hard to degrade them. Of course, over a very long time of exposure to physiologic conditions, they can get a little bit degraded. But most of the removal for the microplastics themselves will be through the macrophages, getting them and phagocyzing them and moving them to the intestine. So yes, these systems are in place, but microplastics are also a different kind of particles. So we really don't know if it is working perfectly or not. What I can say is that in the animals we sampled, you can see here, it was 49 animals, which we collected 242 samples from the internal tissues. The ones, we did not get a lot of information about the animals, unfortunately, but we have the sex, the age, and all this general information. And in the older animals, we did not find a higher concentration of microplastics compared to the younger ones. So we are hoping that there is some kind of mechanism to excrete all these microplastics. But of course, more studies like this, or even in humans, take into account different ages and different exposures, because we have a lot of confounding effects here. Even with the animals, right? If we have a dog that lives indoors, it's different than a dog that is exposed to the air outside. And so to control for all these confounding factors, and then to look at age, maybe we can find something about that. But so far, studies are very limited. Even the ones on human health, as we've seen here, I put some in the beginning, I think. These ones involved very few participants. As you can see here, about 22 participants. This is not enough for a statistical analysis. We really need bigger studies to control for these confounding factors and try to identify the factors leading to higher exposure of microplastics.
Thank you. Other questions for Joana?
There's something in the chat. Yeah.
I might jump in again, if that's okay, Doug, if my internet holds out. I mean, Joana, what you've mentioned is that it looks like with the older animals, there's a clearing out, which is good to hear. But one of the things that I'm pondering on is that given the exponential growth in the use of plastic, I assume that there'll be an exponential growth in our exposure to microplastics. So even though we can clear them out, it's more likely that we'll all receive a higher burden or a higher exposure to microplastics. Have you thought about this?
Well, that is true. I think it depends on our behaviors because, again, there are so many factors involved. Just your cleaning schedule at home can influence a lot what you are exposed to in terms of particles, indoor air, right? If you open your windows, you get more air from the outside, and this air generally is lower in contaminants. And if you clean your floor every day, you're likely to have less microplastics. If you wear mostly organic textiles like cotton, you are likely to be less exposed. So there are so many different factors, but I agree with you. It really depends. If we continue to get more plastics into our lives, we're likely to get more exposed to plastics. And this also includes our foods, because at the moment the trend is to have more processed foods, which usually are packaged in plastics. And so I think in this case, it's very likely to increase the exposure. As I presented somewhere here, I think it was some slides before, just opening the plastic packaging can release a lot of plastics to your food. So, or using the plastic materials. And sometimes these materials are recommended by the agencies looking at commercial kitchens and so on. So I think we have a lot to deal with. The first thing is to know that plastics, we really need these materials. So it's impossible to completely ban plastics from our lives. As we talked about, the electronics use plastics. We cannot live without electronics right now in the modern world. We also need the medical equipment to be made of plastic, so it's disposable. Before plastics, we had people getting infectious diseases through the reusage of materials, even though they were washed and disinfected. Some of it, there were also a little bit of residual contamination, and we were getting people sick because of this. So plastics here, it's very important. In the packaging as well, they are very important to extend the shelf life of food and food safety. But are all plastics this way? Well, maybe we could pick the plastics that are least harmful to us. For instance, the polystyrene, we already know it's difficult to recycle. It can break very easily into microplastics. So maybe not polystyrene, but I'm not an engineer, a chemical engineer to tell you this, of course. So we need different solutions taking into account our modern way of life, keeping up with medicine, food safety, and all that accomplishments we have made, but also only introducing plastics when it's needed, because a lot of times it's just because it's cheaper than the other materials. In that case, maybe we should consider even ourselves individually, if it's not better to spend a little bit more money to buy cotton clothing, if you can find it, than to buy plastics. But it's very hard right now to find something that does not have plastics. So yes, I agree with you. It's very likely that our modern lifestyle will lead to an increased exposure to plastics.
Thank you. And I agree, it's very hard to buy clothes that don't have plastics nowadays.
Very difficult. But even your textiles at home, if you have curtains, if you have rugs, your couch is all made of plastic.
I'm changing it all to wood. Thank you.
Very good, very good.
Any other final questions for Joana? If not, let me once again thank you for a very stimulating talk. It was a really nice talk, and I really appreciate you taking the time to join us today. I should have in my opening also thanked Professor Chen Gu from Nanjing University, who is or has been our regular host for the series, but who wasn't able to be here today with us. Remind you all that all of the lectures are posted on our website, and so if you have missed any of them, or if you just want to go back and depress yourself even further on the state of plastics in the environment, you can go back and listen to the talks again. So thank you all for joining us. Thanks again, Joanna. Hope everyone has a good upcoming holiday season. And as I said, if you have ideas for other lecture series that you would like to see us organize, by all means, get in touch with us and let us know. Thanks to everybody. Bye for now.
Thank you. Happy holidays.